Who here has never used Gramene? Okay. Okay, so because mine is a very general overview, I'll go quickly so that it leaves more time for some of the newer things that have been developed in the last bit with Gramene. One of the things I'd like to get out of the way first is to let you know that we have grown into quite a big partnership with a lot of different organizations. Several of us are at Cold Spring Harbor. A lot of the biological curation goes on at Oregon State University under Pankaj, and those of us at Cold Spring Harbor are under Doreen Ware. We also have partnerships with EBI, with whom we work to load our genomes into the Ensemble framework. We're working with OICR on the plant reactome, and also they help maintain the BioMart tool, working with the American Society for Plant Biology to curate data at the source from authors. We also have funding from USDA-ARS, and so we have quite an established project and a lot of partnerships. Whoops, going the wrong way. Okay, so we have collaborations with the DOE and the major groups releasing the genomes, such as TARE for Arabidopsis and MSU with rice and IRGSP. And so have a lot of, here are the people working on the site. There's about a dozen of us working on informatics and curation, and about half as many PIs at the major organizations that I had mentioned. So not a huge team. What I'd like to tell you about what we've done in the last year is that a lot of what we do is bring in new genomes into the Ensemble structure, so you can view them in the Ensemble genome browser, as well as they get integrated into whole genome alignments, pairwise alignments, and the Compara pipeline that helps you get orthology. Been quite busy with that. A lot from the informatics side, the team at EBI has been working on the highlighting of Go terms in gene trees, which is a new feature. It's very interesting. With every release, we update gene models and EST alignments as needed, or for new genomes that we bring in. We have new variation sets for sorghum and maize. We always stay current with the latest Ensemble browser releases, so we always get the benefits of their improvements and bug fixes. With every release, we have new biomarts for downloading of genes and orthologs and such as that, and the gene trees. And we stay current with all of our ontologies. With each release, we update our ontology database. We have new pathways in both maize and rice that we've been curating. We have redone our markers and sequences database. A lot of people like to come to us for like SSR information, and we've been streamlining that to make it better. The Ensemble team has come up with a new REST-based interface, which is a representational state transfer. It's just a fancy way of saying a very clean, elegant way to programmatically interact with the Ensemble databases. So if what you're looking to do is programmatically get genes and orthology, you can do that through a defined interface now, and you're not limited to just the BioMart tool. That's kind of a beta for us. We have it up on our development site, and we'll be working that in to also expanding it to all the other data types that Gramene hosts outside of Ensemble. And we have new diversity data from Glaberrima, maize, and rice. And under the diversity tool, we also, the tassel development team has released their 4.0 release, which you can use for analyzing our diversity data. I'm going to give you just a brief overview of what the site looks like. This is our home page. And from here, you can see that we have some major data sets that you can just jump right into. Like if you know you want to get into QTLs or pathways, you can just go straight to those sections and start searching and using the tools. If, however, you just generally want to see what we have, you'll probably want to use this bit up here, the quick search. You just put in a search term, and it will help you find something. For instance, one of my favorites, alcohol dehydrogenase. I always search for this. And here I'm looking for it in the taxonomy sorghum because of the wonders of our ontology database. We use the ontology database even for our taxonomies. You can put sorghum, you can put sorghum bicolor, you can put rice or rhiza. They all work because we'll translate them to the official names and do the search behind for you. Let's see, going this way. We have auto suggestions, very 2008 of us. So the search results, when you get this back, you'll see that it breaks it down into the major sections that we have. So if you know you're looking specifically, you want to get into like a genome browser, you would open the top part for genomes. But if instead you were looking for ADH in pathways, then you could just jump down here and open that bit. So here I'm going to look at the protein section. When I open that up, it shows me the 75 proteins that it found. Clicking on one of those will take you to the actual details of the proteins. And it works the same in all the different sections. Here I'm looking for kinase, and you see I have quite a few results. Opening up the pathways, you see that there are actually 4 links here. I can jump right to a pathway, the gene, the reaction, or the enzyme. And that'll take me right to the pathway view if that's what I want to see. If further I wanted to look kinase in just rice, I can do that, and again, just by putting rice up there. And you notice that here now the pathways, it only finds 484, all of which are in rice, in our rice pathways database. And there's all that. A lot of people come to us looking for markers, especially the SSRs in rice. And so we have a nice markers database, and we keep up with the details on all the different marker types that we store. This is what the details look like for an SSR. They have cross-references to the literature. From the literature, you could also find other associations to things like QTLs and maps and such. We keep up with sequences for these, and we obviously also have a BLAST server. Lots of people let us know if the BLAST server goes down, we hear about it immediately. People really like to use our BLAST, and so you can blast a sequence right from the markers database and go right into that, and that helps you get into the genomes as well. Josh is going to talk about in a minute. We keep up with the map positions for a lot of historical genetic maps and QTL maps, deletion maps. And you can get into the CMap view, which is still a venerable tool, I guess. It still works, and a lot of people like to see the maps. You can add maps and get to these comparative views, and that's the CMap tool that Marcella had mentioned that I developed a few years ago. One day we'll replace it with something far, far better, but that takes time. To get into the genomes, I won't cover this too deeply because Josh will be going into it pretty well. But here, if we search for a classic gene, DL1, you see that we find a hit in Arabidopsis and one in rice. And from there, we can go right to the gene summary page and get into the genome browser. And again, Josh will cover that. Another way to get into our genomes is right at the very top. There is a genomes navigation menu, and it lists all the genomes that we have. We're up to, is it 23 or 24 complete genomes? And about 9, I think, chromosome 3 short arms from the wild rices that we've been working with, the OGE project. And so you can just jump right to any of our species from the genome view. Another tool that we offer that's very important is the Biomart tool. You can use this to get, for instance, orthology information or just a list of genes that are related to a GO term or a PO term. This is what the interface looks like. You would choose the plant genes, and you would choose the species of interest. Here I'm showing how to get orthologs for particular rice ID. And I have an LOC ID, and I can put that in there, and it shows me that it finds the gene that I'm talking about. And I tell it that, because I'm filtering on a gene ID, and I tell it that I want to get the gene ID back. And in addition, wrong way, I go over to the attributes now, and I tell it I'm looking for the homologs. And specifically, I'll ask for the homologs in Arabidopsis and Brachypodium, and it works. So I can get this downloaded as an Excel file and do further work on it. Another thing that we work with is the Galaxy tool from Penn State. And so if you start at the Galaxy tool, then you can see that Gramene can be one of the sources, and it takes you to our Biomart tool. And then at the end, in addition to being able to download the data, you can say send to Galaxy, and it'll bring you right back here, and then you can go into the workflows at Galaxy, which is it's a terrific tool. Another thing that we offer is direct MySQL access to our databases. These are mostly our Ensemble core databases. So if you would like to just simply query that without having to download the entire database from our FTP site, which you are also happy to do, you can just use GrameneDB and get to our databases directly. And that's a totally separate database. It's completely outside our firewall, and we don't care if users bring it down with a bunch of queries. So you can go nuts there. We have a DAS server where we allow, where we actually use this to put up custom tracks from our collaborators. And we also have a lot of additional information that you can use to show as alignments on our genomes. And you can also use these in your own external genome browsers. Like, so say you set up gbrowse locally to look at some data that you have. You can use our alignments as well. If you want to keep up with us, Marcella mentioned our Gramene announce list. We also have a news blog, news.gramene.org, and you can subscribe to an RSS feed. Of course, we are on the Facebook, which we just basically is the same stuff as on our news site. If you have any questions, you're welcome to just write us at gramene@gramene.org. We prefer you to use the feedback page, the feedback form, because it feeds right into a bug tracker, and so that we don't ever forget to respond to somebody. We can keep up with the fact that somebody wrote us. And lets us know if you were on the website right where you were, and so we can figure out if there were any problems with the website. And lastly, this is most of us who work on the site.